Let's start apoptosis. So apoptosis literally meaning what? Fall of the cell from the tissue. Fall of the cell from the tissue. For example, this is a tissue. This is what? This is a tissue. And for example, these two cells are fall out and now the tissue become like this. So falling off the cells from the tissue is the literal meaning of the apoptosis. And as I have discussed in the previous lecture, that programmed cell death is seen in apoptosis. But remember, programmed cell death is also seen in apoptosis, necroptosis, pyroptosis, and neutrophilic extracellular trap. If examiner asks that programmed cell death is seen in all these pathways, give me another definition. So your another definition will be energy dependent process. Energy dependent process means that energy depletion take place in apoptosis and the cell lead to a death. So both definition are right. Programmed cell death definition is right, but programmed cell death is seen in all these cases, while the energy dependent process is seen only in apoptosis. So this is a better definition from programmed cell death. And if examiner asks apoptosis is physiological or pathological, so as I have discussed in the last section, that necrosis is always pathological, but apoptosis is both physiological and pathological. And if examiner asks that give me examples of the physiological, so your answer will be that organogenesis due to embryogenesis mean the organ development process, it is physiological. New vessel formation means new vascularization or the killing of the inflammatory cell after completing their function, mean that inflammation begins in the body and now we do not need these inflammatory cells, so these cells die by apoptosis. And the fourth example is elimination of autoreactive cell to prevent immunity. Means the autoreactive cell, those cells that are against our own body system, and we remove those cells by apoptosis. So for examples, these are enough: organogenesis, new vascularization, killing of inflammatory cells, and elimination of autoreactive cell to prevent autoimmunity. Now, if examiner asks that give me the pathological examples, so your answer will be that the chemotherapy or radiotherapy also lead apoptosis and necrosis. Both take place in the chemotherapy or radiotherapy, but now this is pathological. This is not physiological. And the second, glucocorticoid induced apoptosis. Glucocorticoid induced apoptosis mean that after taking this drug and it induces remember, this is a drug which induce apoptosis. It is also an MCQ question in the NEET and AIMS, that which drug induce apoptosis. So your answer will be glucocorticoid. The third is graft versus host disease. The fourth one is viral hepatitis. Council main bodies in viral hepatitis. I will discuss the council main bodies later on in this section. But remember that in viral hepatitis, council main bodies are seen and these are apoptotic bodies. So it is also a pathological example. And the last example is misfolding of protein. Misfolding of protein. So these are the example like pathological. Okay. Now morphology, we will see what is the first sign in the cell injury. Everywhere the first sign in the cell injury is cell swelling. But I have discussed in the previous section that in apoptosis the first sign is cell shrinkage, and it is the first sign. If examiner change the question and he ask about most characteristic feature, so your answer will be chromatin condensation. And if he said that doctor give me a second most important characteristic, so your answer will be cell membrane intact. Cell membrane lost its structure in necrosis. And if he ask about that is there any inflammation, so your answer will be no. And if he asked why there is no inflammation, because the cell membrane intact, the intracellular is not leaking outside. When the new intracellular contents are not leading outside, so it means that there is no inflammation. The fifth morphology is that it is hypereosinophilic. Hypereosinophilic and chromatin condensation. You can see that it is a hypereosinophilic and chromatin condensation. Cytoplasm not specific. if the sixth characteristics you will ask, so you will ask that in the normal cell there are a receptors. These receptors are called phosphatidyl serine receptors. What? Phosphatidyl serine receptors. And these are the lipid receptors, and these are the receptors for eat me signal. That when these receptors move outside, the cell is autophaged by macrophage or any other kind of cell. So these receptors are normally inward, and these receptors are called phosphatidyl serine receptors, and these are the eat me signal. When the cell injury take place, these receptors, instead of looking inside, they are looking outside. When they are looking outside, it means that these receptors are flipped, so now it can easily be killed by any macrophage, and you can see the cell by a lipid dye called annexin V, and annexin V is a marker of apoptosis. The next example is this is a cell and a little bit portion is bleb outside, so it's bleb detached. The bleb is detached. So now when this bleb is detached, it forms its own structure. This bleb is detached. For example, this bleb detached. Now it forms its own structure, and it having its own mitochondria. So when this a cell portion is detached and it forms its own structure, it is called apoptotic body. And what is the definition of the apoptotic body? Membrane bounded structure with organelle, and there may or may not be a nuclear material, nuclear remnants. So this is an example of Conselman body, and if somebody asks that where you can see Conselman body, your answer will be in apoptosis. Now the seventh characteristic is about the scavenger receptor. This is a cell, in this cell having a receptor called a scavenger receptor. This cell having a scavenger receptor. When this scavenger receptor is seen by a phagocytic cell, it leads towards phagocytosis. So these are the seven most important characteristics. Remember cell shrinkage. The most characteristic feature is chromatin condensation. The second one is cell membrane intact. The fourth one is there is no inflammation. The fifth one is hypereosinophilia and chromatin condensation. The second is phosphatidyl serine receptor goes outward. The seven is apoptotic bodies, and the eight is scavenger receptor. Now let's discuss biochemistry of the apoptosis and necrosis. So you will see an enzyme, as we have discussed, the mitochondrial enzymes are present in the apoptosis and lysosomal enzymes in the necrosis. So there is an enzyme called a procaspase. This procaspase is activated into caspase, and this caspase lead towards what? Endonuclease activation, another enzyme activation, and now this enzyme leads to chromatin condensation. So this is a basically a process how chromatin condensation takes place in apoptosis. Procaspase, it is activated into caspase, caspase is activated into endonuclease activation, and endonuclease leads to chromatin condensation. So the functions, what is the function of endonuclease? So endonuclease simple function is nuclear DNA damage in a fragments. In fragments, nuclear DNA damage in a fragment. For example, this is DNA. Now for example, this is DNA, and this DNA, the endonuclease damage into fragments. It is called internucleosomal DNA damage, and the size of the fragment is 180 to 200 base pairs. So can be observed on agarose gel electrophoresis. So you can see that here you can see gel electrophoresis, agarose gel electrophoresis, the biochemistry you can see here. Step ladder pattern is seen in the apoptosis, which is characteristic seen in both apoptosis and necrosis, but it is characteristic for apoptosis. You can see in these the fragments, and it can also be seen in the necrosis. So basically a characteristic feature for apoptosis is step ladder, and for the necrosis is a smearing pattern. We will discuss this smearing pattern also. Let's discuss the mechanism of apoptosis. So apoptosis having two events, one is initiation, one is executional. Initiation further divided into intrinsic pathway and extrinsic pathway. In intrinsic pathway you will see caspase 9, in extrinsic pathway caspase 8 and caspase 10. Caspase 8 you can see in the lower animals and 10 in the humans. So we will discuss with the 9 and 8 in the humans. In the executional pathway you will see in caspase 7, 6 and 3, because it will activate endonuclease and at last chromatin condensation. So this process is basically for chromatin condensation. How chromatin condensation takes place? Through two events: initiation, execution. Initiation, intrinsic pathway, and extrinsic pathway C8, C10 and C9. So let's first discuss about the intrinsic pathway. So we have already known that mitochondria intrinsic pathway is also known as mitochondrial pathway, as mitochondria is the most important organelle. It's a major pathway. But before going to start intrinsic pathway, let's discuss some proto-oncogenes. So proto-oncogenes are divided into pro-apoptotic and anti-apoptotic. Pro-apoptotic means that these genes will induce apoptosis and these genes will prevent apoptosis. These genes will induce apoptosis and these genes will prevent apoptosis. So you can remember by simply anti-apoptotic genes having L in last, except this one. Remember this one, this must not be confused with anti-apoptotic. Except this, all the rest ends with L are anti-apoptotic genes. So BAGS, BAG, BCL, XS and BH3 only BH3 proteins present on the cell surface and stress sensor like BIM, BED, BED, Puma, Noxa. You can remember why BIMLA is bad in bidding for Puma, Noxa. So these all are the pro-apoptotic genes, and BCL2, BCLX and the MCL1. These are the anti-apoptotic genes. So let's start in intrinsic pathway. As we have discussed, BH3 are the proteins present on the cell surface, and these are what? These are the stress proteins. For example, this is mitochondria, and the mitochondria is a double layered, inner layer, outer layer. On the outer layer BCL2 is present, and BCL2 is what? Anti-apoptotic, means it prevent apoptosis. And inside what are present? BAGs and BAG are present. BAGs and BAG are present. So normally when a stimuli comes, there is a BCL2 and it not activates, and it does not activate apoptosis. So when the stress is coming, our cell surface protein are present, which are called BH3 proteins, and these BH3 proteins activates BAGs and BAG and inhibit BCL2. When BCL2 is inhibited, it means that anti-apoptotic function is lost and apoptosis function begins. So now these BH3 proteins inhibit BCL2 and activates BAGs and BAG, and this BAGs and BAG induce what? Mitochondrial permeability. Now the cytochrome C is going outside. The cytochrome C is going outside, and it combines with another molecule in the cytoplasm, which is called apoptosis activating factor 1. And this apoptosis activating factor 1 and cytochrome C, they do what? Activate apoptosome, and these apoptosome activates procaspase 9 to caspase 9. As we have discussed, that procaspase 9 is converted to caspase 9. On the other hand, there is inhibitor of apoptosis. There is inhibitor of apoptosis, and these inhibitor of apoptosis inhibit apoptosome. But how this inhibitor of apoptosis is inhibited? Another molecule from here is leak out, which is called SMAC diablo. SMAC diablo inhibitor of apoptosis molecule, and this cannot inhibit this apoptosome, and the cell is now going towards pro-caspase 9 activation to caspase 9. Now let's discuss extrinsic pathway. Death receptors are present. Death receptors are present on the cell surface mediated pathway. Death receptors are named by tumor necrosis factor receptors and FAS or CD95. When these combines with tumor necrosis factor or any other kind of molecule are combined to these receptors like tumor necrosis factor or FAS or CD95. These receptors activate what? These death receptors, they activate adapter protein, and this adapter protein convert pro-caspase 8R10 to caspase 8R10. So simply extrinsic pathway do this function. And there is another molecule called FLIP, which is anti-apoptotic, and this FLIP is present normally in every kind of cell. Every kind of cell in the viruses HIV inhibit apoptosis in extrinsic pathway. But now this caspase 8 and 10 inhibit this molecule and it cannot perform its function. So this is extrinsic pathway. After extrinsic pathway, now let's discuss executional pathway. Now caspase 7, 6 and 3 is activated, and these activation leads to endonuclease activation, and this endonuclease activation leads to chromatin condensation and apoptosis take place. This was the whole mechanism about apoptosis. Now let's discuss about this PLEP molecule. Now PLEP anti-apoptotic present in the normal cells or viruses, which is inhibited by extrinsic pathway by inhibiting the formation of C8R10. So what is the hallmark of apoptosis activation? So simply caspase activation. But remember, there is an exception. Hallmark of neuronal apoptosis activation is apoptosis inducing factor, no caspase. In neuronal apoptosis, remember for exception and for MCQ question that in neuron, is there any apoptosis by caspase enzymes? So your answer will be no. There is neuronal apoptosis is done by this enzyme, apoptosis inducing factor. Normally, if we relate this apoptosis with a clinico-pathological correlation, so normally what? There is a misfolding of proteins, caspase is activated and it leads to apoptosis. But when caspase cannot perform its function, so misfolding of protein leads to increase in the misfolding of proteins, and it leads towards what? Neurological diseases like Alzheimer's disease, Parkinson's disease and Huntington diseases. Got these points? So this was all about apoptosis. If you need the notes of these lectures, if you are preparing for USMLE or any other kind of exams, you can contact with us and we will provide you these notes, these lectures and USMLE guidance, etc. Anything you like. Have a good day.